Matthew Medeiros. I've known Matt for a few years now, and Matt's currently an assistant professor at the Pacific Biosciences Research Center at the University of Hawaii at Manoa. Matt's group studies the drivers of infectious disease transmission across ecological scales. So from the dynamics within host individuals to even to landscape scales, where he studies the distribution of disease across landscapes. And as we'll see in just a moment, his group combines field, laboratory, mathematical, statistical techniques to address complex questions about disease transmission, and notably to develop evidence-based approaches to mitigate transmission risks, especially in insect-borne diseases. Matt trained as a community ecologist, an evolutionary biologist, and a disease ecologist. He earned his PhD from the University of Missouri at St. Louis, where he worked with the renowned ornithologist and ecologist Bob Ricklefs to study the ecology of avian disease. And after completing postdocs at the University of Brasilia in Brazil and Texas A&M, Matt returned home to Hawaii and joined the faculty at the University of Hawaii in 2016. And since he returned home to Hawaii, Matt has really emerged as a leader in both research and teaching. And among his many leadership positions, he's the chief scientist at INSTAR, which is the core insectary at the University of Hawaii, where they're developing new ways to rear insects such as Hawaiian Drosophila. Matt's also the co-director of CMIT, which is the Center for Microbiome Analysis through Island Knowledge and Investigation. So that organizes microbiome research across the Hawaiian academic community. Matt's an indigenous Hawaiian, and he's developed some really innovative ways to blend Hawaiian culture and traditional knowledge into both his teaching and his research. And hopefully we'll see some of that today in Matt's talk, and we'll have a chance to talk about it more at the tea time, the reception following Matt's talk. All right, with that, let me turn this over to Matt and officially welcome Matt virtually to Oregon. Welcome, Matt.
Awesome, awesome. Thank you so much, Brendan.
You're welcome.
Let me set up this. Okay. Is that working? All right, excellent. Okay, aloha mai people. This is a great opportunity. I'm very excited to share aspects of our lab's work recently. Like Brendan said, my introduction to the idea of symbiosis came through sort of the parasite and the dark side and of the relationship. And since then, I've sort of transitioned to try to understand the breadth of ecological interactions that occur between hosts and the various microbes that sort of live on or within them. And today I'm going to be talking about some of our work on drivers of diversity in the mosquito microbiome. All right, so our lab has become extremely interested in host-associated microbiomes, right? And these are complex ecological communities of microorganisms that really span the tree of life, right? Archaea, bacteria, fungi, protozoa, and viruses are all constituents of host-associated microbiomes. And uniquely, this community of microorganisms interact during the assembly process of the microbiome, not just among themselves, but also with the host organism that they live on or within, right, that they inhabit. And these interactions can range, of course, from parasitism to mutualism, and it can cause disease or promote optimal host functioning. I'm a trained community ecologist, as Brendan mentioned, and I tend to approach microbiomes under the assumption that community ecology provides a really, really great platform for understanding how microbiomes shape in their hosts, right? And I think it provides a lot of good theory, a good body of theory to approach this system. So one of the frameworks that I'm going to sort of reference a lot today is a breakdown of species biodiversity across scales, but I'm going to talk about it in a particular aspect with regard to host-associated microbiomes, right? So at the broadest scale, we have gamma diversity, which is sort of the regional biodiversity, the total number of species and their evenness across a large scale. And then at the local level, we have alpha diversity, which is the amount of species and their evenness on these sort of local assemblages. In the case of a host-associated microbiome in today's talk, I'm going to be talking about alpha diversity as an index to articulate the richness and evenness of individual host microbiomes, okay? And then there, of course, there's beta diversity, which is a little bit more complex. Truth be told, community ecologists are still trying to figure out exactly what beta diversity is and how to measure it. But today I'm going to be talking about beta diversity as the turnover of species between hosts, individual host microbiomes, right? So these individual host microbiomes are distinct subsets from the regional species pool when beta diversity is high. So what do host-associated microbiomes look like across these scales of diversity? Well, in some systems, gamma diversity can be very high. What does that mean? Well, it means that there's a lot of different microbes out there that have the ability to colonize a given type of host, either permanently or in many cases transiently. At the alpha diversity level, again, the diversity of individual host microbiomes, there's a lot of variation in terms of the degree of diversity. For instance, one common paradigm is the fact that vertebrate microbiomes are generally associated with a lot more complexity, a lot higher diversity than invertebrate microbiomes. But one thing that seems to be rather consistent across systems is surprisingly high levels of beta diversity in the microbiomes. That is, there can be a lot of turnover as you move from one host to another host in terms of the composition of their microbiome. And since the composition of the microbiome ties into the types of functions that microbiome can provide the host, there indeed can be mechanisms by which this beta diversity, this turnover, might explain important variation in host phenotypes, even within a population. And I think this really underlies the importance of trying to understand the drivers of beta diversity of host-associated microbiomes. So our lab is especially interested in sort of understanding how different aspects of the environment might modulate beta diversity in host microbiome systems, right? And there are probably numerous mechanisms by which the environment might be linked to beta diversity in host-associated microbiomes. They might have to do with transitions in gamma diversity across the environmental gradient, niche partitioning of the microbes across that gradient, dispersal limitation, even stochastic processes. But one idea that our lab is really interested in testing is the idea that this beta diversity between host microbiotas increases in response to this sort of heterogeneous environmental landscape as a result of a co-selective symbiosis, right? So this idea assumes that a proportion of this turnover is a response to the functionality provided by the microbes to the host. And this is going to sort of underlie a co-selection regime of the microbes on their host and the host on their microbes, right? So specifically, we can look at this diagram and I can explain this a little bit better. Imagine we have this oval, which is supposed to represent a heterogeneous landscape, right? And there's an area demonstrated by a dark green towards the left of the oval. And as you move across it, it gets lighter, and that's supposed to symbolize a change in the environment. And this environment is associated with a regional species pool of microbes that you see underneath it. These microbes provide functions to the host, and the types of functions are more important in certain environments than others. So I've thrown some shading over this, and the microbes that are on the dark end of this green gradient provide functions that are more important in dark green environments, and the ones at the light end provide the functions that are more important in light green environments, right? So if this is playing out, then we expect that hosts might benefit from host-mediated microbe selection, where hosts are associating with microbes that provide functions that promote host fitness in these specific environments, right? But maybe not others. And then the other side of this coin is the microbe-mediated host selection, right? So if microbes are modulators of host performance in the environment, then microbes are providing functions that promote host fitness in some environments, but not others. So if this is occurring across environmental gradients, you might expect to see some proportion of the beta diversity explained by this phenomenon, right? You're going to have this shift in the composition of the microbiome across space simply because some microbes are more important in certain environments. Right? And we have a pretty decent proof of concept with regard to this in an invertebrate system, right? This is Eupremna. It's the Hawaiian bobtail squid. And it has a very specialized symbiosis with Vibrio fischeri, a common marine bacteria. The work of McFall-Knight, Ruby, and others have demonstrated that this squid can set up a series of structural and chemical filters that allow only essentially one functional strain of bioluminescent Vibrio fischeri to colonize the crypts of a specialized organ in the squid, the light organ. So this means that, you know, in the presence of a great diversity of microbes that occupy seawater, only one strain of Vibrio fischeri is allowed to colonize the crypts of the specialized organ, right? And this strain is bioluminescent, right? So it gets into this light organ, it produces light through bioluminescence, and this light promotes countershading in the organism in the water column, and this decreases mortality associated with predation. So here we have host-mediated microbe selection. The squid is engineering this complex bottleneck that whittles down enormous diversity in the water column all the way to basically one strain of one microbe. And you have microbe-mediated host selection because those squids that are associating with that particular strain are suffering less predation in the water column. So one area where this sort of example falls short, though, is the fact that it's hard to pursue across a differential environment because this is a really ecologically obligate symbiosis, meaning that the benefit that this microbe is providing this host is really realized across the whole realized environmental niche of the host organism. To test this idea, we need a species that occurs across various environments where microbes and their different functions may have differential impacts on host performance. And we've chosen to look at this system, the mosquito, right? We work with two mosquitoes in the lab, Culex quinquefasciatus and this guy. This is Aedes albopictus. Aedes albopictus is going to be, I mean, most of the data I'm presenting today focuses squarely on Aedes albopictus. It is a mosquito that was probably native to East Asia, but it is among the world's most invasive species. It has, through globalization and worldwide trade, it has colonized, 6 continents. And in Hawaii, it was introduced in about late 1800s. And it's probably our biggest sort of public health pest mosquito in the state. Now, of course, the importance of mosquito biology to human health is sort of difficult to overstate, right? Several different mosquito species are important vectors of human disease. So for instance, Aedes mosquitoes like Aedes aegypti up here in the upper left corner Aedes albopictus, who I just introduced to you in the previous slide, they are really effective vectors of several human arboviruses, things like dengue virus, Zika virus, and chikungunya virus. And these viruses alone can infect 100 million people per year. In the case of dengue, this can be tens of thousands of deaths globally. In the case of Zika and chikungunya, these infections oftentimes lead to chronic impairment throughout the life of the infected human. We have Culex mosquitoes in the upper right corner. Culex quinquefasciatus here in Hawaii is an important vector of avian malaria, an introduced pathogen that has decimated our Hawaiian avifauna. In North America, it is a competent vector of West Nile virus, a virus that emerged at the start of this century. And globally, it's been a very important vector of filariasis. And then you got the big one, human malaria, which is spread largely by mosquitoes in the genus Anopheles, in the case of human malaria, and it can infect 300 million people per year, leading to almost 1 million deaths per year, much of those deaths biasly affecting children in Africa. So, cumulatively, you're talking about a global burden of morbidity and mortality that is, from a humanitarian perspective, just unacceptable. And this has led to a lot of interest in technologies that can suppress mosquito-borne disease naturally. And one of the really interesting things about the mosquito microbiome is it turns out it's a very powerful modulator of the capacity of mosquitoes to vector these critical diseases, right? And there are numerous pathways by which the microbiome influences the ability of a mosquito to transmit a pathogen. So here I've thrown up an equation of vectorial capacity. It's a measure kind of similar to R naught, and in the age of coronavirus, we're all intimately familiar with what R naught is these days, but this is a measure that estimates the number of infectious mosquitoes that are derived from the whole vector population feeding on a single infectious host for the duration of 1 day. And it's basically a measure of the capacity of a population of vectors to sustain disease transmission. And it's derived, from the same compartmental mathematical models, in this case, describing mosquito-borne disease dynamics that something like R naught would be derived from. And I don't want to get too much into this equation, but it's very simple and it's quite elegant. And I use it a lot to sort of introduce students to math, or introduce biological students to mathematical biology, because it's really easy to understand. But I just want to highlight that there's a couple of really core parameters in the baseline equation. Mosquito to host ratio, the daily feeding probability of a female mosquito, the daily survival of a female mosquito, the incubation period of a pathogen, and the probability of infection given exposure to or taking a blood meal from an infectious host, right? These are some basic and very fundamental parameters in this equation, and each one of these can be modulated through various pathways by the structure and composition of the mosquito microbiome. And in fact, this relationship is so strong that the manipulation of the mosquito microbiome is being proposed as a really serious strategy to mitigate the burden of mosquito-borne disease, right? So what do mosquito microbiomes look like? Well, for everybody out there who studies the very important fungi and viruses, I have bad news for you. There are a lot of very important taxa that undoubtedly make up the mosquito microbiome, but like they are in several other systems, they are severely neglected. So my talk is going to focus on one aspect of the microbiome, the bacteriome. If anybody is interested in the natural history of host-associated microbiomes, there's a lot of work to do in the microbiome and the virome of mosquitoes. So the bacteriome of mosquito microbiomes are dominated, in this case, by proteobacteria, bacteroides, and firmicutes. Most of the diversity is within proteobacteria. The vast majority of this diversity is environmentally acquired, and it is complemented by a few lineages that are vertically transmitted, largely these endosymbionts in the genus Wolbachia. Typically, we're dealing with low alpha diversity. Okay, so the average number of species for an individual mosquito is pretty low, especially compared to vertebrate systems, maybe 5 to 50 taxa per host. Obviously, this number is relating to the sequencing platform you've chosen quite strongly. But in general, all things being equal, we're dealing with more simple communities. But there's an extremely high turnover in the species composition between individual individual hosts microbiomes, right? So a lot of beta diversity. And this is what's so attractive about this system, right? We're interested in beta diversity of host-associated microbiomes. Here we have a very simple, tractable microbiome that we can manipulate, and it shows this pattern of high turnover in species composition between different individuals. So I approach this study of environmental drivers and beta diversity of the microbiome in an absolutely spectacular setting, right? The high islands of Hawaii are extremely environmentally heterogeneous. And for those people who have not visited the state, it's difficult to impress upon them just how heterogeneous and stark these gradients are. When students come into my classes, oftentimes from the mainland, and I want to introduce them to this concept, I talk about an environmental gradient on the scale of the University of Hawaii campus. So as they go from their biology course in the biomed building and they walk down to their flag football class down at the athletic complex in lower campus, that's about a kilometer, maybe a little bit under a kilometer, maybe a 10-minute walk. And I looked at the campus of U of O, and it seems like our campuses are comparable in their scale. So imagine being on one end of the campus and walking clear across the other, and maybe that's a 10 to 20-minute walk. So if you find yourself up by the biomed building at UH Manoa, you're kind of pushed towards the back end of the campus nearest to the mountains. You're going to be in an area that receives about 70 inches of rain a year. Okay, that's about 20 inches more than Eugene receives in an annual year. So we're talking about a very wet environment, as you guys are well aware. Pacific Northwest is a mesic environment, right? So it's a pretty wet place. When my students get to their flag football class and they're out on the field in the athletic facility, they're in an environment that receives about 20 inches of rain a year. Okay, and to get to a place that receives 20 inches of rain a year in Oregon, you gotta hike across some pretty high mountains and get to the east side, and maybe there you can find some place that's quite dry. These gradients are that stark. Literally, you move from a humid rainforest to a coastal desert in mere kilometers. And the reason why has to do with the position of Hawaii in the North Pacific. It's sort of tropical, near subtropical, right there smack dab in the middle. And the height of our volcanoes. We're tall islands and we have high volcanoes. We receive trade winds from the east, and these trade winds are very, very moisture rich. And they hit our islands, and they immediately rise with the altitude, and they cool due to adiabatic processes, right? And as they cool, the air retains less moisture, it forms clouds, and it dumps the moisture, if there's enough of it, as rain. Generally speaking, on that windward slope of the mountain or those mountainous areas. As the air climbs over the ridge and goes down the dry side, the lee side of the island, it is much drier already. It is warming up, so it's capable of holding in more moisture as it moves. And as a result, the lee sides of the island are quite dry, right? So there are places in Hawaii where you can literally walk from a rainforest to a desert, from 300 inches of rain a year to 5 inches of rain a year in a day, actually in an afternoon, in mere kilometers, right? So these are really stark gradients. And when you're talking about the ability of a small little mosquito to disperse, it's pretty neat, because these are gradients that might be experienced by individuals over their lifetime. So we've used these gradients to try to understand how the biodiversity of the mosquito microbiome partitions across the landscape, right? And we've shown that there's predictable changes in composition across this stark landscape scale moisture gradient. So in this particular study, we sampled 10 female Aedes albopictus mosquitoes from 50 different sites across the island of Oahu. And these sites on the island of Oahu were scattered across this landscape scale moisture gradient. Here it's represented by this PC score, where the darker greens are typically these really, really wet and cool environments. The lighter greens being more of those hotter environments, drier environments. We also have about 1 million people on this rock in the middle of the Pacific. So there's some areas that are very heavily urbanized, and then others that are quite natural. So there was also an urbanization gradient nested within this sample. So we went out, we collected at least 10 female mosquitoes from these different places. We obtained their midguts by dissection. We pooled these midguts per site, per collection date, extracted DNA and profiled their microbiome with, sort of standard 16S metabarcoding techniques, very similar to the Earth's microbiome project protocols. And then we developed a series of mixed effects, zero inflated negative binomial regression models to try to understand the response in the relative abundance of individual symbiont taxa to these two landscape gradients using random slopes of the gradient scores for each of the ASV taxa. And we also controlled for spatial autocorrelation among these points in this two-dimensional landscape by adjusting and implementing a spatially structured covariance matrix in these models, right? So we compared the models with information criteria, and we essentially found that the model that included the moisture gradient and a spatially structured covariance significantly outperformed other models, right? In general, the urbanization random slope did not improve model fit, and models that didn't incorporate the spatial structure were very poorly fit to the data. I then plotted this heat map here to sort of demonstrate this effect. So here I have the OTUs on the y-axis, right? So the microbial taxa on the y-axis. And then on the x-axis, I have the 50 sites. And the squares represent the proportion of reads of that OTU in that particular site. Now I've ranked these OTUs and these sites. In the sites, I've ranked them according to our ordination scores from the wettest, coolest environments on the gradient, which are closer to the origin, to the driest ones furthest away, okay? And I've done the same for the OTUs in terms of their response to this gradient based on the conditional means from the random effect. So in general, the conditional means of the random effect suggested that the microbial taxa that are closer to the origin on the y-axis are those that are typically more abundant in wetter environments, and those that are further away from the y-axis are typically those that are more abundant in the dry environments. And you can kind of see how there's this shift as you move up to the upper right corner. There's a shift in the abundance profile of these taxa, and this scales to impact the overall composition of those microbiomes across this gradient. So we wanted to ask whether this type of pattern that we've shown, that other labs have shown in terms of a link between the structure of the microbiome and the host's environment, right? Whether or not it could be related to this co-selective symbiosis hypothesis that I started this talk mentioning, right? So we decided that we'd do a controlled lab experiment, and we wanted to test a few predictions. One is we wanted to test the prediction that the host might select microbes to associate with. And we thought that we could build an experiment, and I'll explain it in a second, where we could see this by matching this prediction in the data, right? Host microbiotas would differ among environments, and they would differ from the sympatric free living environment that host found itself in. And this would suggest that, you know, the host is somehow mediating the assembly of their microbiome from the free living microbiome, that they're somehow adjusting the process. That this isn't just a passive acquisition of niche dimensions that are occurring in the free living microbiome. Then the other side of the coin is the functionality of the symbiosis, right? So we thought that if we saw this pattern, if there are enriched taxa in the host microbiome relative to the free living one, if these hosts, if there's some evidence that they're selecting these special microbes, we thought that these microbes would impact host performance positively in the environment that they're enriched in relative to other environments that they might not be enriched in, okay? So we tested this idea with this large mesocosm experiment. We had 72 mesocosms. Each mesocosm basically was a larval mosquito habitat. And it occurred, you know, I mean, this was done in an environmental chamber in a bunch of sterile T250 milliliter flasks. But we tried to keep this experiment as realistic as possible, right? So we varied two environmental variables in this experiment. One was the temperature regime. So we know temperature is an important modulator of mosquito development. We set a diurnal and nocturnal temperature in the cool treatment to reflect the patterns that are happening sort of at the upper elevational limit of Aedes albopictus's core range in Hawaii. And then we set the warm one to reflect the general dynamics that are occurring at about sea level. We manipulated aquatic chemistry across the other dimension by changing the source of botanical detritus of the larval habitat, that water source for those larvae. We chose two plants to utilize. One is strawberry guava, which is an invasive species, highly allelopathic, and introduced from Central America. And then there's ohia lehua, which is our dominant native rainforest tree. We chose these plants in particular because they form monocultures across the landscape. In the case of strawberry guava, it's highly disturbed, invasive species, monoculture forests. In the case of ohia lehua, it's a natural pattern where our rainforests actually have a tremendously low species diversity in terms of the types of trees that occur there. So it was realistic to us that actually there could be real larval habitat out there on the landscape where the detrital input was dominated by dead leaves from these particular plants. Like this would really happen across landscapes in Hawaii. And then of course we had one treatment that was just pure water. And then finally, we wanted to manipulate dispersal. We wanted to give the free living microbiome in a treatment the ability to move around between mesocosms. So to manipulate dispersal, basically what we did, we take a small sample of the water from every mesocosm in the experiment, and we pooled it. And then in the dispersal treatment, we simply reallocated out that water into those environments. In the no dispersal treatment, we took that same pool, but we double filtered it, and then we reallocate it. So it was getting the same, dose of chemistry, but it wasn't getting the same dose of microbes. And I should say for these botanical infusions that we made to vary aquatic chemistry, we were able to sterilize them with the same sort of filtering strategy, right? So 0.1 micron filter, double sterilized for each of the infusions. Okay. We then added 10 azenic first instar Aedes albopictus larvae to each of these mesocosms. And we added a standardized species pool of microbial symbionts. All of these microbial symbionts, 23 of them in the species pool, were isolated from Aedes albopictus midguts in the field. Okay, so these are ones that at least had the capacity to engage in a symbiosis with Aedes mosquitoes. And the final concentration in the mesocosm was about 10 to 4 bacteria cells per milliliter. So we carried out this experiment. We let it run for about 3 weeks. We allowed the larvae to pupate. When successful, we collected those pupa, we washed them, allowed them to emerge in a sterile environment. We collected the adults when they emerged, extracted DNA, profiled their microbiome using the same sort of standards that I talked about for the other study. And we also did this to the free living microbiome, the water. We took samples across 3 different time points for every mesocosm, so we could make comparisons between the host-associated and free living microbiome. So here's what we saw. We saw 11 symbionts colonize mosquitoes under the conditions of this experiment. So our analysis focuses squarely on these 11 symbionts. We showed that in the free living microbiome, the aquatic microbiome, the 2 environmental gradients, temperature and aquatic chemistry, were powerful modulators of the species responses, changed the composition of the microbiome. We also saw that dispersal, or that the microbiome composition varied slightly with dispersal in a manner that matches sort of a mass effects meta-community model, right? Where poor competitors are maintained in the environment through dispersal from source environments. And as an astute reviewer pointed out to me, that's exactly what we set up to do with the experiment. We wanted to create a scenario where things that would normally go extinct would have the opportunity to sort of persist in certain environments and colonize mosquitoes. And that was in that dispersal treatment. I think what's really interesting about that dispersal result is the fact that it's not present in the host-associated microbiome, right? So there, it didn't matter if we tried to get those microbes in there, they didn't get in there. That seems to indicate, I think, to me, that there's a really strong ecological filter that might be going on. We also found that the composition of the microbiome varied among the sexes. We found that the composition of the host-associated microbiome varied between the 2 environmental gradients, the temperature and the aquatic chemistry. But most significantly is that we found that the patterns that existed, the individual species responses to these environmental gradients in the free-living microbiome were distinct from those in the host-associated microbiome, right? And this was sort of indicating that the host got to be mediating this assembly process somehow. This is not just passive acquisition from the free-living environment or the free-living microbiome. So these differences between sexes were pretty interesting. It's actually really common in the mosquito literature to show differences between males and females in terms of the composition of their microbiome. And it's not surprising. The diet of these mosquitoes differ pretty strongly, right? So females take blood meals and also eat or take sugar meals from plant nectar to sustain their metabolism between blood meals. Adult male mosquitoes only feed on sugar. So, like I said, a lot of different studies have found differences between the sexes. All of them have thought that the mechanisms are related to diet. And the mechanisms that have been suggested in the literature that I can glean are either proximate or ultimate in their explanation. Proximate in the sense that, okay, whatever you last ate, is going to have a really important impact on the composition of your diet. Ultimate in the sense that, maybe male mosquitoes are associating with certain types of bacteria that aid their specialized diet, and likewise females might be associating with certain bacteria that help them, for instance, process a blood meal. Our results are pretty interesting because every one of the mosquitoes we tested was completely unfed. They had never been offered any sort of meal. So this is really indicating, I think, ultimate mechanisms, right? Or at least hinting at the possibility of them, that somehow the sexes are associating with different microbes in the same species pool. So we're really, really interested in sort of testing whether or not that relates to their diet, right? Like, are males associating with certain types of bacteria that they can, bribe with sugar to make them, amino acids or other nutrients that are sort of depauperate in their candy diet, right? Are females associated with microbes that are helping deal with, blood, human blood or vertebrate blood? The next two slides are going to show some of these differences in the different taxa across the treatments between both the host and the free-living microbiome. So here we got the hot treatment and the cool treatment. And you can see the proportion of reeds from each symbiont that came out of the host or came out of the water or the free-living microbiome. And I just want to highlight these cases where we had like significantly different patterns between the host and the water, right? So KPEN1, we can focus in on. That's some sort of Enterobacteriaceae. You can see that in the host microbiome, we have the vast majority of the reeds coming from mosquitoes that emerge from cool water mesocosms. In the case of the free-living environment, it's a lot closer to 50-50. And interestingly enough, KPEN was a microbe we isolated from the very highest extent of Aedes albopictus range in Hawaii. Same thing is happening again with the aquatic infusions. Here we have the pink representing Ohia lehua, the green representing strawberry guava, and the blue representing pure water mesocosms. And I want to key in on these two microbes in particular. So Cry1 is some sort of Chrysiobacterium. And you can see that the vast majority of reeds in the host-associated microbiome came from those that emerged from strawberry guava. But in the free-living microbiome, the vast majority of reeds came from those that came from pure water mesocosms. Pseudomonas had the exact opposite thing going on. The Pseud2, which is a Pseudomonas, was dominant in host-associated microbiomes from those hosts that emerged from sterile water mesocosms. But in the free-living microbiome, it was more common in Ohia lehua and strawberry guava mesocosms, right? So then we thought this is an opportunity to see whether or not these patterns are related to host fitness, right? Is Chrysiobacterium more dominant in host-associated microbiomes from strawberry guava because they somehow improve performance of the larva in those environments? Same thing for Pseudomonas. So we set up a brand new experiment that really paralleled our first experiment, but instead we manipulated the presence of Chrysiobacterium and Pseudomonas, these two strains that we saw these wide differences in terms of across environments and between host and free-living situations, right? So we vary the presence of Chrysio and Pseudomonas, and we replicated mesocosms across strawberry guava and sterile water treatments, right? So the prediction would be that, Chrysiobacterium exposed larvae in strawberry guava are going to do great. maybe it's not that important in sterile water mesocosm. In Pseudomonas, the exact opposite, right? It was enriched in the water. So we would presume that the larvae that are in pure water mesocosms that are exposed to Pseudomonas might be doing fine, and the ones in strawberry guava are indifferent to the presence or absence of Pseudomonas. And that's indeed what we found. So when we looked at least at one really important correlate of host fitness, developmental success, we see that the presence of Chrysobacterium in a strawberry guava mesocosm increased the odds of successful development by 3.2 times. There was no significant effect for Chrysobacterium in pure water. In Pseudomonas, in strawberry guava, there was no effect. But for Pseudomonas, the presence of that bacteria in the species pool of inoculum that we offer these guys resulted in a 4.2 times greater odds of survival. Okay? So this matches sort of that idea that these enriched taxa are associated with environmentally specific patterns of host performance. So where does this leave the hypothesis? Well, I think we have some decent evidence that co-selective symbiosis might be going on here, right? We see predictable changes in the microbiome composition across environments in nature, right? We saw that hosts might be selecting microbes. And we inferred this from patterns where the microbiota is different across environment, but they also differ from the free living environment in that particular mesocosm, right? And we saw that these symbionts that are enriched in certain environments are associated with better performance of the host in those environments, right? So by no means are we done here. I think we've got some pretty interesting results, but really we're very interested in trying to ascertain exactly how widespread this phenomenon is, right? How many other environmental gradients might we see similar dynamics? Did we just get lucky stumbling upon something in this system? I'm not sure. I mean, the other thing is, we can't get into the idea of hosts selecting microbes without getting a pretty good idea of how they're doing it, right? So we'd be really interested in trying to understand the organismal level mechanisms by which these patterns might arise, right? Are they really selecting microbes? If so, how are the hosts doing it, right? And of course, if the microbes are providing increased performance, what functions are underlying that performance? So these things are really interesting to us. So with that being said, I just want to thank my team, sort of acknowledge my graduate students, Priscilla Seaborn, Jason Griffin, Daniel Weber, two extraordinary technicians that I work with, Nicole Yonoishi and Kristen Nakayama, and an undergraduate who worked in our lab on this project, Kahiva Davis, who was great. And then, the development of this project and my ability to understand what's going on has really benefited from conversations with a lot of mentors, Margaret McFall-Nine, Ed Ruby, and of course Brendan, U of O's only. And then, of course, I want to thank the funders. This was all funded by an NIH grant. So with that, I guess that's all I have.
Thanks, Matt.
All right.
We have time for questions as well before we
question. Okay.
Any questions for Matt?
I have a quick.
Feel free to unmute yourself. Go ahead, please.
Okay. So great talk. Thank you so much for this.
Thank you.
You talked about the sex differences, and you said that it's not dependent on their last meal because they weren't fed. So how do you think they're selecting it differently between males and females?
Yeah, I mean, I am not. Here's where I fall short, right? I am not an animal physiologist, and I can only guess at what mechanisms might be available to an invertebrate to make those choices. And this is where collaborations with people who know these things a lot better than I do would be really, really valuable. So I don't know how they're making that selection. As an ecologist, I can just see, right, that we have a species pool and it's standard for all the mosquitoes that are in that mesocosm. And when we look at males and females, they have permitted the colonization and, importantly, the transstadial transmission, right? So these guys were exposed to bacteria during their larval stage. They pupated. Their entire internal organs were completely reshuffled. And then they transstadially transmitted certain microbes, and they did it differentially across the sexes. So I really have no idea how they might achieve that. I do really want to understand how they did, and I certainly want to understand the consequences of that transmission, and how it might impact the diet of, or at least the ability to process the different diets.
Thank you.
Other questions for Matt?
I had a question. Hi.
Yeah, hi.
Yeah, that was great. So one question I was thinking about was for microbiomes for the females, have you or are you aware of any studies that people looked at the impact of like the skin microbiome from like the feeding hosts, like humans, for example? Because I've seen some studies recently in ticks, for example, that actually turns out the skin microbes on humans are important, you know, in ticks during blood feeding. You know, they can damage the tick or other things. So has that been looked at all, or have you guys seen anything to indicate that's important?
So yeah, I know what studies you're talking about. They're really cool. Stuff like that hasn't been done extensively in the mosquito systems. In fact, we really don't really know the environmental origin of a lot of the microbes of the mosquito microbiome, right? We don't know where these things are closely related lineages sort of lie in the environment that the mosquito interacts with. So I actually have a graduate student, and we had this crazy idea that we try to go and figure it out, which meant it was a really fun project because we just go out and we collected a ton of different samples. We imagined ourselves living the life of a mosquito, and we thought, what are the different substrates we're going to interact with? And we went out and sampled it, and we have this huge data set of like 1,000 samples, and we just got some NovaSeq data back not too long ago, and we're really excited to sort of take a dive into it. But one of the things we went out and sampled is the avian host that this bird-biting mosquito is going to interact with. And so we're going to see, hopefully we're going to see if there's some degree of sharing of microbes on the cutaneous surface of the bird and this bird-biting mosquito. So we might have some information about whether or not there's any links there. I do know, and this is a bit off topic, but most of the skin microbiome related research of the host with regard to vectors has been pretty cool. In fact, our skin microbiome is an important modulator of mosquito preference, right? So some of us are getting bit more often by mosquitoes because of the composition and sort of resulting chemistry of our skin microbiome. But this is a little bit of a departure from your original question.
Thanks.
Cool.
Other questions? Peter?
I was wondering if you have any idea or know how we would tell how important, being picked up by mosquitoes and being carried around in their guts would be to, the fitness of their microbes from some of these strains, right?
Right. Yeah, I'm not sure, but I do know of a researcher, and I heard her talk about it briefly. Her name's Carrie Kuhn, and she's at the University of Wisconsin, junior researcher, and she's sort of interested in the evolution of these microbes as they move through symbiosis, right? So we know that these guys are picking up microbes in the environment. They're bacteria, they have very quick generation times, and maybe there's either an acclimation or maybe a full-scale adaptation to symbiosis, right? So the strains in the environment are somehow fundamentally different from the strains in the host. So I think, the mosquito microbiome world has been firmly in the natural history realm, right, up until now. And that's okay with me. I was trained by Bob Ricklefs. He's a natural historian in his heart, and so am I. And I like that space. But now a lot of people are transitioning to sort of understanding some of the more complex evolutionary mechanisms or cellular and molecular mechanisms that produce these symbioses. And I think some of these people with these ideas are going to start finding some interesting things soon.
Any other questions? Well, if questions bubble up, please join us for the tea time. We'll take a break here for 5 minutes or so, and then we'll reconvene at the same Zoom URL for a more wide-ranging discussion with Matt for tea time. But before we go, let's thank Matt one more time for a great talk. Thanks, Matt.